fantastic. So, Sarah.
So Sarah Walker from the MRC Clinical Trials Unit. So I'm fascinated by Rossini too. And my question is really, but not for you, it's for Max and Babak. So what about this issue that effectively you're stopping for futility? I understand you really want to find things that work, but actually sometimes showing things really don't work is very important clinically. You've essentially got a 2 by 2 by 2 factorial. How confident are you? Or is that actually how you did the analysis to show that your initial drape thing didn't work? And is there a way to think about it that in MAMS to balance off the importance of definitively showing things don't work as well as finding things that do work?
You want to go ahead? Yeah, I'll give that a go. So there's a number of questions in there. In your usual fashion, Sarah, you've asked a number of questions. The first is, let's deal with the factorial issue first. We discussed this long and hard, both as a group and with the funding organization. And it's always difficult to know when to jump for a factorial or not. We came down ultimately, and I'm still never sure that this is the right decision, because I don't think you can be, to have a factorial design, but not a factorial analysis. And the reason we did that was we felt that the interventions, the skin, the drape, and the sponge, could potentially interact with each other, either to have a synergistic effect or actually have a negative effect on each other. And so we felt it was important that they were regarded, the combination therapies were regarded themselves as individual therapies. Whether that was the right decision or not, I don't know, but that's where we got to after a lot of discussion. In terms of showing things that don't work, where we got to on that, these were all relatively currently not widely used. They were used by some people, but not widely used. So you really had to show benefit for people to get them to be widely used. So we didn't have to show things really definitively didn't work. We just had to find things that potentially did work. And that's quite a different situation to situations where you've got things that are that widely used that you then need to show definitively people should stop using them. There's no reason now that the arms that we've stopped that people will go on to use them and say you should have got more data. So it was enough to show actually, interestingly, the arms that we have stopped, at least on the first stage, the results seem to suggest, if anything, there was harm from some of the interventions. So actually the likelihood that they actually worked was very, very small. So it was pretty clear they didn't work. But
Sarah, has Max answered all your questions? Excellent.
Yes, it is, very much. It's not enough, no. I don't know whether you want to add to any of that. No.
Okay. I think the gentleman over there in the middle. Yeah. Again, just say your name and your institution.
Just shout if you.
Yeah.
Sanjay, respiratory physician, Oxford. Thank you. I had a question for the Octopus trial. I noticed it's a placebo trial, and I don't think Stampede was. Very relevant for respiratory and neurology trials because our outcomes are very symptom dependent. EDSS has always been the primary outcome for phase 3 trials for MS, and annualized exacerbation rates are very important for COPD and asthma trials. Two things that the FDA, MHRA always demand is placebo and a replicate trial before you can have license. So how does MAMS fit into studies that first need a placebo? I don't know if the next generation of intervention in Octopus are going to be injectable or infusions or subcut or IM. And two, if you do one really big trial, who's doing the replicate?
So I guess there are a couple of questions there. So everyone's on standard of care, which includes disease-modifying treatment, progressive disease-modifying treatment. Then randomized onto that are control or active arm A or active arm B. What was the next point?
It was the placebo question.
Yeah.
So this is a tricky issue, isn't it, of placebo, and the way we've got round it, both in the MND SMART trial and with the Octopus trial, which are the two trials in this area which have got off the ground, is use over-encapsulation, or actually in the MND SMART situation, liquid preparations, but all the liquids at least appear and taste the same. We've managed to do that with 2 active arms to start with. As you indicate, and this is always a trouble when you have people who know about trials in the audience, isn't it? They ask difficult questions. As we go forward, there will be an issue about, as you say, if you get an injectable or it gets more complex, you've got 4 or 5 arms, you can't necessarily over-encapsulate them all in the same way. That's a challenging problem with a placebo added to the control arm issue. We haven't solved that. There are a number of potential solutions, none of which is necessarily ideal, but we will have to solve that problem. The thing with these trials we learned is don't solve a problem unless you absolutely have to. Acknowledge that you're going to have to solve it, but work out the timeline that you're going to have to solve it. So hopefully it will be solved before you start your respiratory trial. Yeah.
Going down, if then there's 10-year license, 15-year license, how are you going to get these 2 collaborators into your placebo control platform trials? I think these 2 questions key for.
Well, yeah, in theory it can be done, but we can talk about the details of it. But you're right, the challenge comes when you add more arms and or the mode of administration of the new agent or a new intervention is different. Then what do you do about that placebo? And there are potentially a number of solutions, and you just need to work through the pros and cons of those. And they're challenging to work through, but actually worthwhile, because again, the prize at the end of it is really large.
Okay. I think there's one there, and then we'll move to the left side. Yeah.
Where's that for, sorry? Thank you. Richard Hayes, London School of Hygiene and Tropical Medicine. Firstly, this has been such an interesting morning. Thank you so much for organising this. Max and colleagues really appreciated it. I think I only have one question. As a statistician, I was just reflecting on how I would analyse some of the data from some of those trials with arms leaving and joining and so on. So just to compare, Rossini, very straightforward because the outcome is very soon after enrollment. It's within 30 days. So at each stage of the trial, you know, the patients are recruited, they get their outcome and they've gone. So you're moving on to the next stage. It's a new start and everything is comparable. It's in parallel. But then going back to that very complex champion diagram, Max, where you've got so many arms and they're leaving and joining, and then you've apparently got a control arm that like goes along for 20 years. So you've got, and that's a case where I think with prostate cancer, probably it's long-term follow-up of patients. So they're not recruited and out in 30 days, as in Rossini, they're carrying on with their follow-up. So I was just reflecting how you would analyze that, because there'd be issues of comparability, comparing patients who'd been randomized 15 years ago with ones who had just been randomized now because they're in a new arm. I hope my question is clear. That's quite a tricky and complex analysis, I think.
So, go ahead, Max. Yeah.
It's definitely not me, this one.
Like the previous question, I think there were a few questions within that. In that
yeah,
question.
there are a few questions in there. So a basic principle is you only compare each arm against its contemporaneous or concurrent control. So you retain the integrity of the randomized comparison. And one way of thinking about this is for each arm, it's just a 2-arm randomized trial, and you can ignore the other arms for that comparison. So then it becomes quite simple. What adds a little bit to the complexity is when you change either, when you add an arm or you change the control arm and the research arm to add a therapy which is shown to be working. And one way of thinking about that is what you've done is close the old trial and you started a new trial either with an additional arm, So if you'd had previously 4 arms, you've now got 4 research arms, you've got 5 research arms. And you just start a new trial and you analyze that almost like a meta-analytic way before and after you made that change. So it's 2 trials. You don't present it like that, but that's a way of thinking about it. And similarly, when you change the control arm and the research arm to add a treatment, you go back to the factorial type random argument that before and after you've made that change, you analyze in a factorial way. Concurrently randomized patients are compared directly, and the only change you're making between the research arm, or the only comparison you're making with the research arm and the control arm, is the difference in that, which is the research arm intervention.
I don't have anything to add.
Yeah.
But we have a few questions also.
From
that,
Menti.
no.
Oh, okay.
No, not on that. I don't have anything to add.
I might just turn to this side of the audience. I think there's a question there. Yeah. No, it's a... Can you just speak up a touch? Just can't quite. So I think the question is particularly on oncology when there's remission, does that cause you any problem? I mean, your time to events.
Yeah, could you just repeat the question? I mean, I didn't hear, I was on a flight yesterday, so I didn't hear the question so well. So would you repeat the question? You
said first of all, it's good to be in trials, I think was your first point. And secondly, was there a particular issue with remission of people causing difficulty with analysis?
As a common endpoint in oncology,
okay.
That's not death. So would that cause trouble?
Okay, so if I understood the question, this is about the role of the intermediate outcome and does it cause a challenge in what that might be and how you might use it and it not being a surrogate. So the argument in these trials is that the intermediate outcome is such that to see an effect on the primary outcome, such as overall survival or death, to see an improvement in that, you need to see an improvement in the intermediate outcome. So to see an improvement in overall survival with a cancer treatment, you need to see an improvement in disease recurrence or reduction of progression. But just because you see an improvement in disease recurrence doesn't mean to say you will definitely see an improvement in survival. That's why it doesn't need to be a surrogate, and you use it to be as a way of screening out potentially inactive and ineffective treatments, but don't claim a winner until you've got the result of the primary outcome. Does that answer your question?
Well, yeah, I think. Are you really saying that the primary endpoint of death is the key thing?
Yes, it is.
Yeah.
Absolutely. You're only using that as a way of saying, is it worthwhile randomizing further patients to evaluate this therapy further? Or actually, should we just call it a day because it's unlikely to be effective because it's not very active?
Okay, on the Menti Do you have some questions coming in?
Questions
Yeah, Babak, far away.
The first one is asking me. I first repeat the question. Can you comment on the column on power in the table for red trial? Is that for each pairwise comparison or overall? And following that, is 80% power enough, 80% or 70%? That's a very good question, because when you're to answer the first question, what is the power? The power was calculated under one treatment, one intervention being under the target effect size and the rest as null. And you're right, the power changes with the assumed target effect size, so like any other trial. And you need, for this trial, we assume different target effect sizes and calculate the power for assuming the target effect size for the given, for one comparison, two comparison, three comparison, and compared all the designs, all these scenarios with the full MAMS design and two-arm trials. Regarding the second question, the second part, is 80% or 70%, 78% power enough? I think that depends on your funding, how much you want to invest, as any other trial. So I hope I answered that question.
So we've got a couple more minutes.
We have more questions.
I'm sure, and we've got some in the audience. So do you have a burning one there that you must be answered now? Everything must be answered, but
we have a few on randomization. Max, would you like to go on this? Yeah,
I'll leave it to the chair to decide.
yeah, I think let's that's yeah, over there. Yeah, no, you've had a question already. So earlier, I remember everything. Yeah. Say who you are and where you're from. Yeah.
Thank you for a really interesting morning. Claire Waddington from Imperial College. So my sort of take-home message of these trials is that essentially it allows you to look at multiple different things at the same time. And my question is, can you look at multiple endpoints at the same time? So if you take something like this surgical site infection, as the surgeon cares about the infection at day 30, as an infectious diseases physician, I care about whether you're generating antimicrobial resistance at the same time. Is it possible to do that?
That's for you. Yeah. Rossini. No, I think we'll go to the surgeon here, because you have to do all the, you do all that stuff.
I mean, I think it's got to be powered on something, hasn't it? Like any other trial design, you've got to pick your primary. We are collecting a lot of other information, and it's such big trials that we're doing with 6, 8, 10,000 patients that we're going to get a lot of other useful information. The control arm skin prep is fascinating, because there's 5 things that we use, and we might be able to unpick that question better than anyone else has been able to, given that we weren't randomizing that, but we were collecting that. I think your other questions about other endpoints to collect, not in the traditional way. I don't know if that might help.
So yeah, I'll give that a go. In principle, so if you think of some of these designs as a smart way of doing two-arm trials, it's not the only way to think about it, but it's one way of thinking about it. Then in theory, there's no reason why you, at least potentially, can't use a different primary outcome measure and a different intermediate outcome measure for different arms. No reason, at least in principle, why not. You'd have to make that argument to why that primary outcome measure is most important for that intervention in that disease. But in principle, no reason why not. We haven't addressed that issue directly. We've discussed it. Well, I may have discussed it just internally, but we have discussed it. And in principle, there's no reason why not.
Okay, so I think it's 12:15, so 45 minutes for lunch. Come back here at 1 o'clock. Please come and answer further questions to all of the panels as we go. And thank you very much for the morning. Thank you.
We have so many questions. What we might do is a webinar system. Can we answer your meeting?